All right. Hello and welcome. So I hope um everyone's having a good day. Um, I hope that only one person sees this video, but we're back at it talking about DNA replication today. Um, this is the last hurrah. I'm super excited. Um, let's get a move on because I've got a family who's already like this.
Okay, people, tomorrow morning, 10 a.m., Santa's coming to town.
Santa! Oh my God! Santa here? I know him. I know him.
Yeah. So we've previously talked about a lot of different topics. Chemistry 1, it's kind of like kindergarten where you get basics, but chemistry at this level, we start to see a lot of applications. And pretty much the bridge between clinical and um, biological functions. So we've talked about alcohols, um, ethers, thiols, alcohol halide or alkyl halides, aldehydes, ketone esters, carboxylic acids, amides, amines, last but not least, lipids, carbohydrates, proteins, viruses. That's where we get into the biological um, interesting aspects. And let me tell you, I will not get confused when someone asks about saturated versus unsaturated fats. Anyways, so today's topic we're going to be talking about is DNA replication and genes. Yeah, not those genes. Um, genes. So a gene is a portion of DNA molecule that's responsible for the synthesis of a single protein. And an individual chromosome is actually composed of many genes. Humans have 46 chromosomes, 23 pairs. So, of course, we cannot talk about DNA unless we put a typical cell up. Again, this is like the classic picture of the cell with all of those different organelles listed there, and we'll pay attention more specifically to the nucleus where the DNA is housed. But remember, this is eukaryotic. Prokaryotic cell, it functions a little bit different. That DNA is hanging out in the cytoplasm, doesn't have that nucleus. So again, how important this is, why we study chemistry. Chemical structure and cellular function are inseparable. So, for example, like in a grossly meaning large like term, like anatomy. If we don't know anatomy, and how in the world are we going to be able to even diagnose something? So it hurts here in my calf, right? And when I plantar flex. So we can get clued in that the common things being common, it's likely a gastrocnemius strain, calf, what are you doing, things like that nature. So when we talk about function, we are talking about, I guess, the way things work, and you won't know how things work until you know what structures come of them. Macromolecules. This is thanks to my microbiome class, actually. I thought this infographic was really, really good because ultimately we talk about cellular level. We've talked about all of these actually in this class. Sugars, when we talk about larger units of the cell, polysaccharides, fatty acids, we talk about fats, lipids, and the membranes, that lipid bilayer, right? Amino acids, obviously code for proteins. We've been hounded in that. Nucleotides and nucleic acids. We're going to be getting into that here in a bit. So, a couple things. Let's go back. Yes, nucleoside versus nucleotide. This is interesting because they're easily, it's literally a one letter difference. And so I wanted to kind of highlight which was what, just so we're on the same page about that when we're talking about components of the DNA and RNA, kind of. So nucleoside is not quite all of it. And by that meaning, we'll get into that. So that sugar and the nitrogen base. So nucleotide means all of it, and that's actually the addition of that phosphate group. So we'll look at that here in a second. So nitrogen containing base. So we have a monosaccharide technically as the base, and then adding that phosphate group is making it a nucleotide. And that, I believe, is adenine. So we'll also talk about this here in the next slide. Adenine, yeah. So that is actually the name of the purine base there. Base, all the different bases. We have the sugar, and then we'll talk about 5' and 3' here in a second. But anyway, so adenine, guanine, uracil when we're talking about RNA instead of T in the DNA for thymine and cytosine. So when we talk about that, we're talking about just the base. When we're talking about the nucleotide, or the actual whole entire component, so remember nucleoside is just these two, and when we add that phosphate group, it's going to be a nucleotide. It's going to be like, for example, adenosine instead of adenine or guanosine. So let's get on with the next slide. Check for understanding. Pretty much we've gone over nucleotide, DNA, essentially what DNA is, RNA is, both polymers. The monosaccharide is ribose versus deoxyribose, taking that one of those oxy groups off. And big thing is the T is in DNA and U is in RNA. Again, so this is a good example. The phosphate and sugar backbone, we all know, and the extra component. So a nucleotide contains the backbone and the sugar phosphate groups, and all polynucleotides contain the same sugar and phosphate backbone. So the identity and order of the bases distinguish one polynucleotide from another. Polynucleotide has one free phosphate group at the 5' end, and a nucleotide has a free OH group at the 3' end. This is what we're talking about. So beginning at the 5' end, we have a CATG or cytosine, adenine, thymine, and guanine. So this is all 3 prime, 5 prime. This is all what that looks like. Okay, so we will get into this real quick about the bonds. There are 2 essentially between hydrogen bonds between adenine and thymine, and 3 actually between guanine and cytosine. And that just has to deal with the principles of the structure of each. There's one more bond between C and G. And you'll also see like the sugar phosphate backbone lies on the outside of the helix, and the bases lie on the inside, perpendicular to the axis of the helix. So the 2 strands of DNA run opposite directions. So you'll see opposite directions with a 3 to 5 and a 5 to 3 prime going opposite ways. And the purine base is always 1 strand away from a purine base on 1 strand always has hydrogen bonds with a pyrimide base. So we know that the purines, well, look at this slide again. The purines are the adenine and guanine and pyrimidines, cytosine and thymine, and uracil for RNA, will never be together. It will always be a purine matched up with the C or T, and always a G paired up with a C and a T, and never these 2 together. So now that we know the structure and how it's composed, we'll talk about the actual replication part. So replication is a process of which DNA makes a copy of itself when cell divides. Transcription is the ordered synthesis of RNA from DNA, and in this process, the genetic information is stored in DNA and passed to RNA, and translation is the synthesis of proteins from RNA. So in that process, the genetic message is contained in RNA for the specific amino acids to make a protein, essentially. So when we get into this, we'll talk about enzymes a little later, but essentially that DNA opens up, and there is a leading strand when we duplicate and a lagging strand. So it actually opens up by enzymes called helicase, right? So and actually I'm working ahead of myself. So we also want to remember that this DNA strands can be up to like 6 feet long in the real world. So we need to remember that like this DNA is so tightly kinked and coiled and folded and all of those things, all up in a nucleus that is like microscopic. And so we have to essentially use enzymes to help us do all these things, all the steps of DNA replication. So topoisomerases, I think, is a blanket term for the unkinking and uncoiling for that DNA. Once it's unkinked in that section, right, we're going to do strand separation. So remember, the two strands need to be opened so that the other enzymes can bring the proper nucleotides in the spot. So helicases are those enzymes that unzips the DNA. And this is what I think is the most easiest part. Helicase, remember, the DNA is in a helix, so you unzip the helix with the helicases. Next is elongation, essentially, and this is when the DNA is actually replicated. So we have enzymes, polymerases. There's more specific ones if we want to get into the woods about it, but essentially we have enzymes putting those bases, I guess the pairs or the complementary bases to each one, like the A and T and A and G or C, or T and G or C. So, and then termination. There wasn't a lot on this, but know that when the copying is complete, there's a couple other enzymes, surprise, to finish this again. So there comes a protein to protect the unmatched DNA for a little bit. And then also there's an enzyme that comes back and is the glue, essentially, to put everything together. So we talked about, let's go back to the leading and lagging strand. And the reason why there's a leading and lagging strand is because, so we have a 3 and 5 prime, and the way the order goes always is 5 to 3. So the upper one up here, this is the leading strand because it will be going from 5 to 3. It'll just be going in the order. That's why this arrow is going like that. And in the lagging strand, it still has to go 5 to 3, but it's in the opposite direction. So they have to work in chunks, right? So it'll have to go 1, 2, 3, 4, and then jump back, and then 1, 2, 3, 4. So it's almost like that. If you ever in gym class, it reminds me of that Indian run where the last person from the end of the line, the whole class gets in a line and they're running, and the last person in the line has to sprint to get to the very front of the line. So it's kind of restarting all of that again. And those are Awasaki segments. So when you see that, that's what they mean. Those are a part of the lagging strand when we're talking about DNA replication. All right. And I think that is all for today. But if you have any questions or anything like that, feel free to shoot an email or something. But I've always wanted to do this. It was one of my favorite parts of being in conferences, being lectured by fairly high highly renowned physicians, and they shared a little piece about their family at the end of their lecture. And it was really sweet to do. So, yes, this is my family. My husband and 2 kids. Hayden is going to be 3 in March, and Maddie just turned 5. So we're busy, busy, busy with these kids, and so excited to get back to the holiday season. So, blessings to you and your family, and good luck in a journey called life. Bye bye.